Hi, my name is Filippa Brugliera and I'm Head of Research and Development at Cann. This facility is our Bundoora R&D facility, and this is where we do most of the R&D on the cultivation and production stages of the company. It's a critical part of identifying new cultivars for us to commercialise at Bundoora. One of the main projects that we run with Agriculture Victoria, one of our partners, is the accelerated breeding program where we aim to produce novel elite cultivars for commercialisation. And so Agriculture Victoria are experts in the field of molecular breeding. And so what we do what we call the phenotyping in this facility. The phenotyping is really looking at the physical traits of the plant growing. So we look at height, we look at bud yield, we look at various bud aesthetics. And what Agriculture Victoria do as part of that project is they take samples of the plant, they sequence the DNA, that's called genotyping. They also analyse the cannabinoids and terpenes that the flowers produce, that's called chemotyping. And they also, using the DNA sequence, they produce phylogenetic trees, and that's kind of like an ancestry lineage of our lines. We put all of that data together and produce a predictive formula. And what that allows us to do is when we do cross various lines and we produce thousands of seeds, we're able to look at the DNA of each of those plants that originate from the seed and enable us to sort of select the best performers just based on the DNA sequence. So we are at that point with Agriculture Victoria. It's been a very successful partnership. We also have projects with La Trobe University. So La Trobe University are one of our main partners. We're part of the Medicinal Agricultural Hub, which is an ARC-funded project. And La Trobe University run many projects around, specifically around answering questions on growing cannabis and around utilising Cann's genetics. And so one of the projects that's been very successful is the tissue culture program. And the tissue culture program is critical to Cann's success in that it enables us to have a kind of a backup or a risk mitigation strategy to hold our different genetics in a sterile environment. Tissue culture is basically, the production of small plantlets in a sterile environment. So we have those as backup, and also we're working on producing tissue culture plant material in our sterile conditions that then will be transported to Bundoora to go into their production facility as potential mother plants. And so it's a way of producing clean stock for the commercial facility.